Hi guys, you are welcome to my YouTube channel. Today we are going to be discussing about DNA and RNA extraction. Introduction. So DNA and RNA extraction are both procedures that are involved in isolating and purifying nucleic acids from cells. And this help us to get DNA of high quality, DNA and RNA of high quality, which can be used in biotechnology, molecular biology, epidemiology, and genomic demonstration. So what are the methods that are used in DNA extraction or RNA extraction? Generally, nucleic acid extraction. So there are basically three techniques or methods that are used in extracting nucleic acid. We have organic extraction protocol, which is the popularly known as phenolchloroform extraction. We have non-organic method, which actually uses the process of salting out and treatment with proteinase K. And we have adsorption method, which is the solid phase method, which employ the use of silica gel membrane. So this third method is used by many extraction kits that are available, commercially available. They use this method of silica gel membrane. So for the purpose of this video, we are going to be adopting the adsorption method, which is the silica gel membrane method. So the uh, nucleic acid extraction kits that we are going to be demonstrating it today is the Ron-May virus nucleic acid extraction kit. And this Ron-May virus nucleic extraction kit has its own advantage over the other ones. You can both extract DNA and RNA using this extraction kit. So what is the principle? And this kit, it uses a unique buffer system and a centrifugal adsorption column that specifically binds virus DNA and RNA. So the centrifugal absorption columns can efficiently and specifically adsorb viral DNA or RNA and remove impurities and proteins as much as possible. So what is the now sample requirement? A sample can be extracted immediately after collection. That is to say, when you collect your samples, you can immediately carry out the process of DNA extraction. Or if you collect the sample and you are not going to carry out the DNA extraction at that moment, maybe because you collected the sample in the field and you need to bring it to the laboratory, so you may not be able to carry out the DNA extraction at that moment. So you can store the sample refrigerated at 2 to 8 degrees Celsius overnight, so that the following day you can carry out the extraction protocol. Or if you are intending to carry out the extraction but on a long time basis, so you have to store it for a very long time before you carry out the DNA extraction. So you need to store it in at -20 or to -80 degrees Celsius. So this is best way you can store it for a longer period of time before you come back to do the DNA extraction. So how do you prepare your sample? And you should know that your sample may either be animal or plant tissue. So if it is animal or plant tissue, you have to ground it with physiological buffer or phosphate buffer saline, PBS. And here, if this plant tissue, for plant tissue, you know, you need to use hash method in grounding them. You need to use mechanical method to ground the tissue very well, because hard plant tissue, some plants, they are very hard. So the tissue is very hard. You need to apply mechanical force to it in order to ground it effectively. Then after that, you add your buffer and you centrifuge so that you collect the supernatant. So it's the supernatant that you are going to use for your DNA extraction. So if it is a serum, plasma, or other body fluids or other samples, so you can carry out your DNA extraction directly using the sample. So if the sample is not up to 200 microliter, you can add phosphate buffer saline or physiological saline to make it up to 200 microliters. So this protocol means for this protocol, you need to have a sample that is at least 200 microliter. So for virtually all the samples you are going to carry out your DNA extractions, you will see that they are going to be more than 200 microliter. So for eventually you are not having up to 200 microliter, you need to top it off with PBS or physiological saline to 200 microliter so that you carry out your DNA or RNA extraction. So what are the procedure or the instructions that are under this uh, extraction kit? So the first thing to do, you already prepare your sample, and what you are going to do is to pipette 200 microliter of the sample into a clean 1.5 ml centrifuge tube. And after that, you add 200 microliter of lysis solution, you vortex, and you shake it for 10 seconds. So what is this lysis buffer or lysis solution? This lysis solution, you can have cell lysis buffer or tissue lysis buffer. They are detergent solution that are used for lysing cells. And this lysis buffer, they are made up of buffering salts, example like tris HCl, or ionic salt like sodium chloride. And these buffering salts or ionic salts, they are help in maintaining the pH. And they can also have detergents such as Triton X100 or SDS, that is to say sodium dodoside sulfate. So these are the components of this lysis buffer, and their function is to lyse the cells. And vortexing helps in rapid mixing. So it helps in mixing your sample with the lysis buffer so that you have good lysis of the cells, and you have the cell membrane being broken up so that you have your nucleus exposed, because you are carrying out DNA and or RNA extraction, so you know your nucleic acids, they are in the nucleus. So you break the membrane with lysis buffer so that you have your nucleic acid exposed. So, and after you have done that, you are going to allow that to stand for 10 minutes at room temperature. Then after you allow it to stand for 10 minutes at room temperature so that you have your lysis occurring, then you are going to add 200 microliter of absolute ethanol. You vortex and shake for 10 seconds. So the essence of adding absolute ethanol is a ethanol that is around 99 to 100% uh, uh, ethanol. So it is ethanol that do not have any water in it, okay? And the function of adding absolute ethanol is that it helps in precipitating the DNA, because DNA is not soluble in alcohol. So it tends, when you add alcohol or ethanol to the sample, it helps the DNA to clump together so that it aggregates and you have the precipitates of the DNA. Okay, that is the function of adding this absolute ethanol. Then when you do that, you are going to carry out your now absorption column. You place it into a collection tube. The absorption column, you place it inside collection tube, and you transfer the above liquid into the absorption column. Then you centrifuge at 8,000 RPM, revolution per minute, for 1 minute, and you discard the filtrate. So this adsorption column is, in other words, you can call it binding column. It is coated with silica membrane, and this silica membrane has affinity for the nucleic acid that you are extracting. So when you place the adsorption column inside your collection tube, you transfer the content into the absorption column, you centrifuge, and when you centrifuge, you will have a flow through, and that flow through you are going to discard it as is your filtrate. You don't need that because your binding column has silica gel which has affinity for your nucleic acid. So the nucleic acid is going to be trapped in the what, in the silica membrane inside the adsorption column. So you discard the filtrate which has your flow through has membrane of the cell that was lysed, and also other cellular materials that you don't want. So the next thing to do is to add 600 microliter of wash buffer 1 to the absorption column, and you centrifuge at 8,000 revolution per minute for 1 minute, and you discard the filtrate. So the essence, what is this now wash buffer? Now, this wash buffer, they are of two types. There is wash buffer 1 and wash buffer 2. So this wash buffer, they help in washing the column. They remove salt and other contaminants prior to DNA elution. So they help in ensuring that you have high purity DNA, and it helps in keeping the DNA bound to the silica membrane. Okay, so and this wash buffer 1, wash buffer 2, they are actually, they differ in ionic strength and the concentration of the ethanol that is inside them. You remember before you start the extraction, when you open your kit newly, you will see that the wash buffer has to be, you have to add alcohol because it comes concentrated. So you need to add ethanol to it in order to carry out the extraction procedure correctly. So it means your wash buffer was initially concentrated, so it was ethanol was added to it in order to what, to prepare it for the washing steps. So then the next step is to add, you return, you have discarded the filtrate. You remember you are washing down the column, dislodging all unwanted contaminants and making the DNA to be bound to the column. You understand that. Then the next step is to add 600 microliter of wash buffer 2 to the absorption column, and you centrifuge at 8,000 revolution per minute for 1 minute, and you also discard the flow through. So the filtrate, we have explained that the wash buffer 1 and 2, they differ in their ionic strength and the ethanol concentration. So they helps in what, getting high purity DNA. So when you do that, you are going to now return the adsorption column to the collection tube, and here you are carrying out the process of drying, because now drying will help in removing traces of the wash buffers. So when you return the collection tube at the absorption column inside the collection tube, and you centrifuge it at 8,000 revolution per minute for 2 minutes, and you discard the collection tube. You are going to discard the filtrate and the collection tube together, retaining your adsorption column. You remember your adsorption column has your the nucleic acid bound to the silica membrane. So you discard the collection tube and the filtrate together. So the next step is elution step. And in this elution step, you are going to place your absorption column inside RNase or dearness-free 1.5 mil centrifuge tube. So you collect a new 1.5 mil centrifuge tube, which is RNase or DNase-free. Then you place the absorption column inside and you add 50 microliter of elution solution directly onto the center of the absorption column. The reason why you are going to do that, you are going to add it directly, not by the side of the tube, is because when you add it directly, it will what helps in what making a maximum contact with your what nucleic acid, because this elution solution is helping in what dislodging your nucleic acid from the what silica membrane. So you know that DNA is soluble in low ionic strength, and these low ionic strengths are usually Te buffer or nucleus-free water. So your elution buffer is nucleus-free water, which helps in dislodging the nucleic acid from the silica membrane. So you add this 50 microliter elution buffer directly onto the center of the of the column, so that you have what maximum interaction. So you leave it at room temperature for 1 minute, and that will allow a maximum interaction. Here, you can actually play around the protocol. You can, if you need like a highly concentrated DNA eluted, so you can reduce the volume of the elution buffer. You can add, instead of 50, to add 35 microliter, so that you have what highly what concentrated DNA eluted. And also you can add the incubation time, okay, to 5 minutes, so that you have maximum interaction with the elution solution, so that it helps dislodging a maximum number of the DNA molecules or RNA molecules before you actually elute. So the last step of the elution is to centrifuge at 8,000 revolutions per minute for 1 minute, and you discard the absorption column. Then you have your nucleic acid extracted, and you are going to store it at minus 20 degrees Celsius. Here also you can play around the protocol, because you need a maximum amount of your nucleic acid. You can centrifuge at maximum speed, let's say around 15,000 revolutions per minute, and you can increase the time to let's say 5 minutes, so that you have what your DNA or RNA are collected in much higher yield. So then after, okay, this is now the operation flow. This is like the summary of how the DNA extraction protocol take place. You have your sample, you added 200 microliter of your sample, then and you remember you added lysis buffer, and after adding lysis buffer, you incubate, you shake and you incubate to allow it to disintegrate your cells, okay? Then after that, you add absolute ethanol, which which helps in precipitating the nucleic acid, and you transfer the content to the absorption column, you remember, and you spin. After spinning, you discard the flow through, you added wash buffer 1, you spin, you discard the flow through, you add wash buffer 2, you spin, okay, you discard the flow through, then you spin the empty columns for drying, and you added 50 microliter of elution buffer to dislodge your DNA or RNA, and you allow it for maximum interaction. Then you also centrifuge, and you collected your nucleic acid. So after collecting nucleic acid, there is one last step that you are supposed to do. You need to know the concentration of your nucleic acid, the concentration of your what? Nucleic acid. So how do you now know the concentration of nucleic acid? Is by carrying out nucleic acid quantification. So, yeah, nucleic acid quantification. Now, for DNA and RNA quantification is a process that is used to determine the concentration of your DNA or RNA in a sample. So why is it important to do DNA quantification? is used to determine the purity of the DNA and also if there are presence of contaminants in the biological sample during the DNA extraction process. So how do you do that? You carry out this quantification process using a special spectrophotometer called the nanodrop spectrophotometer. So you are going to collect like 1 microliter of your sample, 1 microliter of your nucleic acid, and you place it, and it's going to the machine. The nanodrop spectrophotometer is going to read the concentration for you. So for highly pure DNA, there is range at which you have absorbance of the nucleic acid and absorbance of protein. You are taking the ratio of the absorbance maxima of nucleic acid and absorbance maxima of protein. So when you have A260 over A280 ratio of approximately 1.8, this is generally accepted as pure for DNA, while a ratio of approximately 2.0 is generally accepted as pure for RNA. So this brings us to the end of this lecture video, and we're going to stop here. Kindly thank you for watching. If you have not yet subscribed, kindly subscribe to our channel so that we and put on notification bell so that whenever we upload a new video, it's going to be notified to you. Thank you. Bye bye. See you.